Hello everybody, and welcome to this GEN webinar. It's entitled Target Enrichment Technology Optimized for Analysis of SNPs, Mutations, and Copy Number Changes in 509 Cancer Gene Targets. Targeted resequencing is one of the fastest growing applications for next generation sequencing technology. The ultimate goal is to look for causative mutations within discrete genomic loci to enhance diagnostic and therapeutic development. Today's webinar will focus on a novel NGS target enrichment workflow. Specifically, our speakers are going to talk about the Single Primer Enrichment Technology, or SPET, S-P-E-T, for the simultaneous targeted analysis of multiple types of variations. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as moderator. But now, let's meet our webinar presenters. Dr. Luke Sherlin is Director of Technical Support at NuGen Technologies. Luke will provide an overview as well as details about the SPET system. Dr. Stephanie Welga works as Lead Bioinformatics Scientist at NuGen. Stephanie will discuss applications and the different types of informative data that can be obtained using Single Primer Enrichment Technology. After the panelists make their presentations, there will be a question and answer segment. Feel free to send in a question for our panelists at any time during the webinar. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible. So if everyone's ready, let's get going. Dr. Luke Sherlin will be our lead presenter. Luke, the floor is yours.
Thank you, John, and thanks for the opportunity to discuss NuGen's products with the audience here at the webinar. Much of the effort in targeted resequencing today is focused on studying cancer and inherited disease. The aberrations most important in this testing, diagnosis, and treatment include translocation events, including fusions, copy number alterations, and single nucleotide variations. Researchers continue to use independent approaches and techniques in order to interrogate these aberrations. In the case of gene fusions or translocations, this includes fluorescence in situ hybridization, or FISH. Copy number alterations are most often measured using arrays, and single nucleotide variations, such as SNPs, utilize arrays or targeted resequencing approaches. So obtaining a complete understanding of all the events contributing to cancer, for example, can be complex and quite costly, requiring not only expertise in multiple assays, but can also consume limited sample in a number of different sample-hungry workflows. The NuGen Ovation Target Enrichment System makes available a single low-input workflow that can provide information on multiple aberrations, including mutations, CNVs, and gene fusions. This approach can greatly accelerate the time and significantly reduce the effort required to get to biologically and clinically relevant data. The NuGen Ovation Target Enrichment family of products offers a high degree of flexibility in interrogating genes or other regions of interest. Much of today's discussion is going to focus on the latest addition to this family, which is a 509 gene Ovation Cancer Panel 2.0. This system bears all the important features of our previous systems, including the ability to interrogate exon mutations and gene-level copy number variation in a single sequencing run. The system enables low inputs, which will allow you to make the most of your precious samples and can be used with either fresh frozen or formalin-fixed samples. The new panel expands on the original cancer panel, adding to the original 344 cancer genes from NuGen's Ovation Cancer Panel 1.0. And in addition to these, including genes included in the Foundation 1 cancer panel, as well as 23 genes that have previously been shown to have copy number variations associated with cancer. Importantly, if the content of the 509 gene cancer panel requires some additional content, or if your studies are focused on a smaller region of the genome, NuGen offers fast and flexible custom designs with a turnaround time of approximately 3 weeks. The table shows examples of a few custom designs that have been created for customers and collaborators. As you can see, there's a broad range of target sizes from a single exon at 5 kilobases to hundreds of genes, upwards of 3.5 megabases. Customers have had success with designs exceeding 20 megabases. The table also shows the variety of different species, sample types, and applications and readouts that have been applied using the Ovation Target Enrichment Systems. So at this point, we'll switch the focus a little bit onto the workflow itself and the mechanism of action. Here's the workflow for the Ovation Target Enrichment System, our simple single-tube solution for targeted resequencing. Input for the system is 10 to 500 nanograms of fragmented DNA, which goes into end repair, ligation of the forward adapters, and a bead cleanup step. This is followed by typically an overnight hybridization step, which is followed by primer extension, bead purification, and a final enrichment. After a final bead purification, the target enriched library is ready for sequencing. The overall workflow takes about 24 hours, starting on the afternoon of day 1 and ending on the morning of day 2. And as you can see, we have systems that are configured for multiplexing up to 96 samples, and the sequencing uses standard Illumina primers, so no custom primers are required. Next, we can take a little bit closer look at the molecular mechanism, and this features our Single Primer Enrichment Technology, or SPET, which is a unique approach for creating libraries for targeted resequencing. As you can see, the goal of any targeted resequencing is to focus the sequencing efforts on specific regions of interest to obtain readouts, including, as we've mentioned before, single nucleotide variants, and indels, rearrangements, or copy number alterations. In this example, we'll consider a target of a single exon for simplicity. In this design approach, we target regions that are adjacent to the region of interest, the region that we're going to call the probe landing zone. And you can see this as the red dotted lines in the inset. In this case, we're designing primers to interrogate this single exon. However, in most designs, we will be designing many probes to hundreds or even thousands of exon targets. In the case of long contiguous regions or large exons, we employ a tiling approach, whereby we have more than just 2 probes interrogating a particular exon, but actually have additional probes across the entire region of interest to ensure that we have even coverage across the entire target. In this case, we do have 2 probe landing zones, 1 on each strand. And if we look more closely at this region of interest, we can see how this can work in your hands. The first step of the process is fragmentation to achieve the optimal fragment size for sequencing. And as you can see, we've taken our large genomic DNA and fragmented it down to regions that are approximately 500 base pairs in length. As you can also see, we have a number of different fragments. In some cases, we have an intact target, and in other cases, we only have a fragment of that target. So now we'll get to our first enzymatic step of the process. And this first enzymatic step is ligation of an indexed forward adapter to each strand of each of these molecules. And you can see that we have ligation of a forward adapter to both the top and the bottom strand in each one of these. In some cases, we're ligating to fragments that are of no interest. And in other cases, we do have a target region where we will see enrichment taking place. If we take a closer look at the adapter structure, what we can see is that the adapter has not only an 8-base index, which allows for multiplexing, but also has what we term an N6 tag. And this N6 is a random hexamer that is also read during the index read that allows one to mark and identify PCR duplicates. The way that this works is that each of these will have a random N6 sequence. And so we associate this random sequence with a unique ligation event that's taking place. Because the fragmentation is random, we will expect to have a random distribution of different start sites throughout these fragments. And by associating the random tag with the random start site, we're able to identify a unique ligation event taking place to a unique insert. And any of these fragments that contain multiple copies of the random tag and the same start site, we expect to only arise from a PCR enrichment event. So as we move on to the next step of the mechanism, this is where we actually have our single primer enrichment technology taking place. And of course, by single primer, what we mean is that each one of these independent molecules is going to be initially enriched using just a single primer from a single direction. And the way that we do this then is to have the target-specific probes. As we mentioned, we have a probe landing zone that's adjacent to this particular target. And these also include some additional sequence that allows us to take these libraries onto sequencing on an Illumina sequencer. And if we look a little bit more closely at the structure of this, we can see that not only do we have a sequence-specific target, or sorry, a sequence-specific probe that's going to hybridize to our probe landing zone, but we also have a sequence that is the reverse flow cell adapter sequence that will allow us to have a complete library molecule for sequencing. And now what we're going to be doing is adding these sequence-specific probes to the mixture and hybridizing them to these denatured molecules. After the overnight hybridization takes place, we can see that the probes themselves have annealed in a sequence-specific manner only to the probe landing zones. And the next step is to perform the single primer extension reaction. And this single primer extension reaction then is going to create a full-length library molecule. And the extension is only going to be taking place where we have a sequence-specific hybridization event taking place. And as you can see, after the extension takes place, we end up with full-length library molecules that contain a forward flow cell adapter that includes the index as well as the N6 tag, and our reverse flow cell adapter. And that's now a complete amplifiable library molecule. And all we have left at this stage is to take these enriched molecules, and we can see this on the next slide. We'll amplify only these molecules that contain the target sequence and the forward and reverse flow cell adapter. And after amplification, now we have an amplified sequencing library that is ready to go on an Illumina sequencer. So at this stage, I'm going to hand off to my colleague, Dr. Stephanie Welga, who will share with you some of the mutation and copy number data that we've generated using the Ovation Target Enrichment System.
Great. Thanks, Luke, for the introduction of the technology. Just quickly, I want to remind everyone that if there are any questions, please remember to submit your questions at the bottom of your screen for the Q&A at the end of the webinar. So now I will spend some time going through some performance data for the Ovation Target Enrichment System with genomic DNA inputs. So on this first slide here, I'm showing some sequencing metrics for the various range of probe sets that we've developed both in-house and also for some of our customers. On the low end, you can see that we have a custom panel that is 35 kilobases in target size, and on the higher end, another custom panel with a target region of 7.1 megabases in size. And what you can see is that regardless of size of the panel, the assay performs essentially the same. So we have high percentage of reads that align to the genome, a high percentage of those reads that are on target, and the uniformity is relatively stable across those different target size ranges. We've yet to see any real limitations yet on our target size. As Luke mentioned, we've also now done a 22 megabase panel that we've seen success with. You'll see in bold there that I've also put the metrics for our new Ovation Cancer Panel 2.0 next to the metrics for our previous Cancer Panel 1.0. And even with the added size of approximately a megabase and a half, we're still seeing similar metrics. So in order to display the accuracy of variant detection with this assay, we've applied the Ovation Cancer Panel 2.0 to 100 nanograms genomic DNA of a HapMap sample NA12878. These enriched libraries were sequenced on a MiSeq, and then after some pre-processing of the sequencing data, including some quality trimming, probe trimming, alignment to the human genome, and PCR duplicate removal, we used the Broad Institute's GATK to call variants. And I'll briefly mention here that very soon we will be releasing a NuGen base app that will take care of these initial pre-processing steps for you given the raw sequencing data as input. So look for that on base app page very soon. So similar to what I showed on the previous slide, at the top here we see that this particular HapMap data set also has a high alignment and on-target rate, as well as a good uniformity and a low PCR duplicate rate. So once we got a set of variant calls out of GATK, we compared the target bases within the target panel that have more than 20x read coverage to an annotation file from NIST which is a high confidence set of variants for this HapMap sample. And we can download this from Genome in a Bottle. The bottom table summarizes the concordance results for single nucleotide polymorphisms. And you'll see that despite the shallow sequencing depth for this particular sample, we're able to accurately call 1,040 true positives with only 1 false positive and 2 false negatives, displaying that we are in fact getting a very high concordance of known mutations. When you think about the samples you might have, not all your samples are perfectly pure, such as a tumor that might be a mixed population of cells. In that case, it's important to know the limit of SNP detection of this assay. So what percentage of DNA can we still identify mutations? So to model that, we've mixed 2 HapMap samples, again NA12878, into NA19238 at various ratios. And then they're enriched samples with the Ovation Cancer Panel. So each of the libraries were sequenced and then analyzed for allelic frequencies of 14 SNP locations that were unique to the HapMap sample NA12878. So as you can see at the center of the plot, when we have a 50% mixture of these 2 samples, you see the presence of each of those 14 SNPs at 50%, where 50% of the allele is derived from the 12878 sample and 50% is derived from the other sample. As we step down and mix only 20% of the 12878 sample with 80% of the other, we see close to 20% allelic frequency of the 14 SNPs. And then in the zoom-in box in the corner there, you can see that we are accurately identifying allelic frequencies down to even the 5% mixture. And even still, at the 1% mixture, we are able to identify most of the variants. So we're very confident that in mixed samples, you will be able to identify at least down to 5% consistently with this system. So that summarized some of our performance data for SNP detection. Now I will talk a little bit about some performance data for copy number detection with this system. So to test our ability to accurately detect various copy number levels, we've used back clones for 2 different genes, EGFR and KIT. These are 2 genes that have been shown previously to have copy number variation. And we added these constructs to promega male genomic DNA. Promega male is a mix of several individuals, so it very nicely serves as a copy number reference genome, where all genes are assumed to be approximately 2 copies, or 2N. So these back clones were spiked in to achieve 3, 4, 6, 10, and then 20 copies in this promega male genomic DNA. And these values were verified by qPCR. The libraries were then enriched with the Ovation Cancer Panel 2.0 and then sequenced on a MiSeq. Because of the consistent performance of our probes, we are able to apply a very simple probe-based read counting method to compute copy number values from the processed sequencing data, and then compare that to the normal promega male counts. So as shown on the graphs here on the slide, for both EGFR on the left and also KIT on the right, we are able to replicate the expected copy numbers quite well, from 3 copies all the way to 20 copies. Shown on the next slide here, in addition to detecting copy number gains, we have the ability to detect significant copy number losses. Again, to model this, we applied the Ovation Cancer Panel 2.0 to promega male genomic DNA and also to HapMap female NA12878 genomic DNA. Across the x-axis of this plot, there are 509 target genes in the Ovation Cancer Panel 2.0. They're listed in chromosomal order from chromosome 1 on the left to chromosome X on the right. And on the y-axis is the log ratio of the measured copy number levels in the male compared to the female. So on the far right, you'll notice in red that the genes on the X chromosome show a statistically significant copy number loss as expected when comparing a male to a female sample, displaying that we are accurately detecting copy number gains as well as these observed copy number losses. To further test the accuracy of our copy number measurements, we've also applied the Ovation Cancer Panel 2.0 to 100 nanograms genomic DNA from 4 cancer reference samples that we've obtained from HorizonDx. So these particular reference samples have varied copy number levels measured by qPCR for 4 genes. And those genes are ERBB2, also known as HER2, MET, EGFR, and Aurora kinase A. So as an example, let's look at sample number 1 at the top of the table shown here on this slide. In the third column, you'll see that this sample has an expected large copy number gain for ERBB2, and it's close to 30 copies. There's no copy number change for MET expected, as it's close to 2. There's a minor copy number gain for EGFR, and a moderate copy number gain for Aurora kinase A. In the fourth column, we list the copy number levels as measured by the sequencing data compared to a normal copy number sample. And you'll see that we get very similar results to what you would expect based on the qPCR. Additionally, with a student's t-test, we can also attribute a p-value associated with the computed copy number change. So all the genes that were noted to have a copy number change did have a significant p-value associated with that change. So ERBB2, EGFR, and Aurora kinase A have significant p-values, while genes that remained at a normal 2 copies showed no significant copy number change, as was expected. So in this case, MET, we see no significant p-value there. So on the right, we can correlate across all 4 samples and all 4 genes the qPCR copy numbers to the sequencing data copy numbers. And we see a very nice correlation of the copy number measurements, which we're quite happy with. So far, I've shown in-house performance results with our Ovation Target Enrichment System. And now I'll show you some customer data that they have allowed us to share, where they were able to very nicely replicate known SNPs and copy number changes in clinical samples. So this particular customer has a custom designed panel targeting a focused set of genes, and they applied this custom panel to 32 patient-derived tumor and normal FFPE samples. Because this is customer data, we were provided the data without complete sample information, so we don't know the actual tumor content of the samples or if the tumor is polyclonal or monoclonal. But what we do know and have is a set of validated 12 copy number changes and 15 SNPs that were identified with an orthogonal method, the Foundation 1 screen, and for 9 of the 32 samples. Shown on this first slide here, the results, these are the results for the cross-validated SNPs with the variant and genomic location of the variant listed on the left, the sample and the expected mutation listed on the middle columns, and finally the occurrence of bases within the customer's Ovation Target Enrichment sequencing data at that particular SNP location. So as an example, we can look at the first one in the table where we're expecting to find in the ATM gene a G to an E mutation in sample 2. And we see on the right that we are picking up this mutation in sample 2 with about 60% A's at this location. So again, because the sample information was blinded to us, we don't know the expected allelic frequency. We just know that the mutation is present in this particular sample, and we were able to confirm that. Encouragingly, for the SNPs listed, we were able to confirm detection of the expected SNP, as well as identify other SNPs for further validation within the samples. We also ran copy number analysis for this same customer data set, and shown in the table on this slide are the 12 cross-validated copy number changes. On the left 2 columns, we see the gene and the sample with the expected copy number change. And in the right 2 columns, we list the copy number as measured by the Ovation Target Enrichment data, along with the p-value significance associated with the copy number change. Again, the copy number is computed, as I've briefly described, using a simple counting approach followed by comparison to the reference normal copy number sample. So taking the first line of the table again as an example, in CDKN2A, we expected a loss in sample number 8. And we've measured now 0.53 copy number loss in the data for sample number 8, and that measurement is statistically significant change denoted in the final column. In the second example, we also expected a copy number loss in sample number 11 for the same gene, and we again see a loss with the measured copy number of 1.5. Although this particular copy number change does not pass our stringent significance test, as noted in the final column, but we are still detecting this loss. As you can see in these 2 examples, there's only a reduced copy number here, so we're not looking at a complete elimination of the gene, and that's because we expect in any clinically derived sample that the tumor content will be under 100%. So this will lead to some background level for the gene due to the nature of the mixed sample. Alternatively, in some cases, we could be observing a heterozygous deletion, and in which case there would still exist a single copy of the gene within that particular sample. So again, because the samples were blinded to us, we don't know the exact nature of the tumor content, but we are able to detect the changes. So within these clinical FFPE samples, we are able to validate these events, and we're also able to validate additional candidate copy number changes in all 32 of the samples. So on the next slide, we can look at a representation of this. So in this plot, each of the 32 samples is shown as a separate point along the x-axis, and the y-axis shows the copy number for the MCL1 gene as measured by the Ovation Target Enrichment System. As expected, we pick up a statistically significant gain of MCL1 in sample 4, colored in blue on the left, along with another statistically significant copy number gain of MCL1 in the experimental sample number 26. And that sample has no pre-existing copy number information, so this can be used as a candidate to be further validated. And then you'll also notice here that across the other samples of the data set, there are no statistically significant changes of the MCL1 gene, and as you can see, the copy number is very close to 2 across all the other samples. On the next slide here, there's one more example where we have the level of FGFR2 gene plotted for each of the 32 samples. And as expected, we pick up a statistically significant gain in sample number 7, colored in blue, along with some additional statistically significant copy number losses of FGFR2 in some of the experimental samples shown in red. So again, these additional changes can be used by the customer as candidates for further copy number validation. So that summarized performance metrics for our Ovation Target Enrichment System with genomic DNA inputs. And now before ending, I want to quickly mention our Ovation Target Enrichment System as applied to RNA inputs for the detection of gene fusion events. This is something that we just published this summer in PLOS One. Our Ovation Gene Fusion panel targets 500 genes known previously to be involved in gene fusion events, either by a handful of public databases or literature searches. So for the probe design here, unlike DNA, we don't have introns flanking our exon targets. So for the design of our probes for the RNA inputs, we've taken a different approach where instead the probes are placed inside the exons at the edges facing out towards the exon-exon junction. So shown on this slide are two examples of how we can make gene fusion identification. In the case on the top, we've designed probes to the exons of both genes of the fusion event. The blue arrows on the top strand can be extended in the direction of the arrow across the exon-exon junction and into the red gene. So this creates a forward read that would come from the red gene and a reverse read that would come from the blue gene in order to identify the fusion event. So additionally, the red arrows on the other strand of the cDNA can also be extended across the exon-exon junction into the blue gene, creating a forward read that would come from the blue gene and a reverse read that would come from the red gene. So you'll also be able to detect the fusion that way as well. In the second case at the bottom, we have only designed probes to target the exons of the blue gene. And you can see, though, that the blue probes pointing towards the gray gene will still be extended across the exon-exon junction and into the gray gene. So because of this, we're also able, with this technology, to detect novel gene fusion events to genes outside of those included in our fusion panel. This next slide is actually figure 2 from the recent publication, and it shows why using targeted RNA sequencing for fusion detection is so important. So across the bottom are 4 gene fusion events that we've identified in Universal Human Reference UHR RNA. And along the y-axis, we're looking at the number of sequencing reads that support that fusion event. In blue, we have a targeted sequencing experiment sequenced with only 1.6 million reads. And in green, we have another targeted sequencing experiment sequenced to greater depth at 8.7 million reads. In red is a publicly available data set that was a 125 million paired end standard RNA sequencing library. So if you look at the first example fusion, BCR-ABL on the left, even at 1.6 million reads for our targeted sequencing library, we're able to identify the same number of fusion supporting reads as in the 125 million untargeted library. In the second fusion, the NUP214XKR3 fusion, you'll notice that at 1.6 million targeted reads, we identify fewer than the untargeted experiment. Although what's important to note here is that we've only designed probes to the NUP214 gene, and we did not have any probes targeting the XKR3 gene. So you can see that we can still easily identify gene fusion events with only one gene targeted. So in the last 2 fusions listed on the graph on the right, we were unable to identify any sequencing reads in the untargeted data sets. So even at 125 million RNA-seq reads, we were unable to detect these gene fusions that we actually were able to detect with a fraction of the targeted reads using our Ovation Target Enrichment System. finally, this schematic displays all possible known fusion events that can be detected with our Ovation fusion panel. So I'll end there and summarize what Luke and I have talked about. The Ovation Target Enrichment System is a very streamlined workflow that can generate target-enriched libraries from fresh as well as FFPE samples with as little as 10 nanograms genomic DNA or RNA. Along with the Cancer Panel 2.0 and the Fusion panel that I've mentioned, we also have a fast and flexible custom probe design to target specific mutations that you might be interested in. With the sequencing data derived from these assays, you have the ability to detect a variety of genomic and also transcriptomic aberrations. And for the case of gene fusions, we've shown that we can detect more gene fusions with our system than with an untargeted RNA sequencing library. Finally, our custom panels are done very quickly and are delivered within 3 weeks of the initial design approval. If you'd like to get more information about Nugen products, you can visit our website at www.nugen.com. So we'd like to thank you for your attention, and I'll hand it back over to John now, and we're happy to take any questions.
Thank you, Stephanie, and thank you, Luke. You both provided a concise and highly detailed description of the SPET system. I'm sure all the members of today's audience who are involved with NGS research learned much about how to improve their workflows. Indeed, I think you really struck a chord with them based on some very interesting questions we received. So let's take a look at the first one. First question for you. Can you adopt this entire workflow to Ion Torrent platform?
Yes, we can. Our current commercial products, the CMV panel, the Cancer Panel 2.0 that Stephanie talked about today, is designed specifically for the Illumina platform. However, we are working with customers to adopt custom panels and other panels for Ion Torrent as well. And we'd encourage you to get in contact with us here at Nugen, and we can work together and put a panel together for Ion Torrent as well.
Great, Luke, thank you. And Luke, I've successfully used the SPET assay for SNV testing, and I'm interested in looking at changes in splicing that may be caused by some of the mutations I've found. Probably meant SNP. Is it possible to use the fusion panel design to look for splicing variations?
So short answer to this is yes. We didn't talk at length about our fusion systems. However, the way that we design probes to detect fusions at exon-exon junctions lends itself very well to detecting splice variants as well, as essentially it's the same events that are being interrogated. So as I mentioned, the system itself does lend itself well to detection of splice variants along with fusions, copy number, and SNPs.
Thank you. Stephanie, how many reads do I need for copy number detection?
A good question, and something that we've been asked a lot. Really, it's going to depend on the target size and so the size of your panel, the number of probes that you're dealing with. But as an example, so for this Cancer Panel 2.0, we have 509 genes, and the data that we've been showing here has been quite shallow actually, and we're still getting very accurate copy number calls. So we're looking at 1.5 to 2 million reads that we're still getting really good copy number results.
Okay. And Stephanie, can I design probes for gene fusion detection and SNP detection?
Yeah, absolutely. So we have a really flexible probe design process, and so if that's something that you're looking to do, we can definitely accommodate that. Basically, we would just do the fusion type design with probes at the exon-exon junctions of your gene targets, and then provided a list of target SNPs, we can add additional probes within the exons that will also capture the SNPs of interest. So yes.
And Luke, can I design to a custom reference genome like a virus or bacterium?
Yes. So the system is set up in such a way that we can design to any target genome as long as we have an accurate reference to work from. So we can work from bacteria, we can work from virus, or virtually any other organism that has a reliable reference genome.
And Stephanie, is there a software package available to compute the copy number values?
Yes, so there is a publicly available tool that we've applied to our data sets before, and we've had some success with it. It's called Contra, C-O-N-T-R-A. You can download it at sourceforge.net, and we've had success with it. But what I've shown today is not actually using Contra. It's using this simple probe-based counting method, and that's something that we describe in our user guide for our customers.
And Luke, how many gene fusions can be detected with the gene fusion panel?
So this is a difficult question to answer, simply because the system itself is really set up to detect both known and unknown fusions. And so the way that the probes are designed to interrogate across a fusion junction means that we can interrogate a fusion where we have two known partners as well as the fusion of a known gene and an unknown gene. And so while we have 500 genes in our commercial fusion panel, there really isn't a limit, so to speak, to the number of fusions or different fusions that can be detected with the fusion panel.
And Luke, is it possible to measure translocation and integration events using the SPET approach?
Yes, we certainly can do that as well. Much like the fusion approach, we can design probes in such a way that they will sequence regions where an integration event has taken place. And so whether we know where that integration takes place or not, as long as we know what the integration vector sequence is, we can design probes in such a way that we'll discover that information. And so this can be done for transposable elements within a particular genome or foreign vectors like viruses.
And Stephanie, what is the genomic resolution on measuring copy number changes?
Sure. So currently we have the system working very well for gene-level resolution of copy number values, although we are doing work in trying to get copy number values for portions of genes. But yeah, currently it's gene-level resolution.
Thank you, Stephanie. Well, unfortunately, we've run out of time, but please note that this webinar will be archived for 6 months on our website, www.genangnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends. I want to say thanks again to the panel for the outstanding presentations, and thank you to our audience for your attention and for your very thoughtful questions about various topics brought up during the webinar. And thank you to Nugent Technologies, whose support made this webinar possible. Bye for now.